Hello AP Biology students, let's go over the 9th section in Unit 6. And in this section, we're going to talk about a topic called gene mutations. Remember from our last section, mutations are any change in the DNA sequence. And a point mutation is going to be a change where a single nucleotide is changed. We also call these gene mutations because they can affect how a gene produces a protein. Now, the first type of point mutation is called a substitution mutation. This is when one base pair is replaced with a different base pair. For example, you can see T is replaced with C. Remember that DNA is going to code for a protein, and how we determine the amino acid structure or sequence of that protein is by looking at codons. Codons are, again, those three nucleotides that occur in a row in DNA or RNA. Now, if we substitute even just one of these nucleotides, it could have a major change on that protein. There are three different types of how substitution mutations can change a protein. The first is a missense mutation. This occurs when a mutation changes the amino acid in that sequence. So you can see in our regular codon, TTC is going to be transcribed into AAG, which again translates into lysine. During a missense mutation, this lysine is going to be changed to a different amino acid. Within missense mutations, there are two different types, conservative and non-conservative. And no, we're not talking about politics. You can see here in a conservative mutation, the property of lysine being basic remains the same, even though the structure of the amino acid is different. In a non-conservative mutation, we are going to change the property where you can see lysine here is basic and threonine here is polar. So again, conservative missense mutations don't change the amino acid property, while non-conservative mutations do. A nonsense mutation occurs when the mutation changes the amino acid to a stop codon. At this point, the ribosome is going to stop translating, which means the protein is not going to be fully made. As you can imagine, depending about where this stop codon is inserted, this is going to drastically change the structure of a protein. If this new stop codon is towards the beginning of a gene, you won't make much of that protein, which is really going to change that protein structure and function. I've been using this meme for years for nonsense mutations, and it is just amazing. Our last type of point mutation is called a silent mutation. This occurs when the mutation doesn't change the amino acid from the original DNA sequence. So you can see in our original sequence, TAT produces tyrosine, however, TAC also produces tyrosine. So since the amino acid's not changed, it's known as a silent mutation. These essentially won't change the structure of a protein since the same amino acid sequence is going to be there. Again, top quality meme here. A kind of different grouping of point mutations known as frame shift mutations, and this occurs when one or more nucleotides are either going to be added or removed. This completely changes the reading sequence or the codon sequence of the gene. You can see our original sequence of amino acids is totally changed when you just move that reading frame one nucleotide to the right. And like you can probably predict, this is going to drastically change the structure and function of that protein. One of the main ways that a frame shift mutation can occur is through an insertion or a deletion mutation, where we just add or remove one DNA nucleotide. Again, I'm going to end on a perfect meme. Frame shift mutations, yeah, they really mess up proteins.